Hello everyone, welcome to Biotech Multiverse. Today we will discuss about production of vaccines. In last video we were discuss about vaccine. What is vaccine? What is the role of vaccine in our body, etc. And today we will discuss the production of vaccine. How vaccines are produced in large amount for everyone. Let's start. In previous classes, we were discussed about recombinant DNA technology, the tools of recombinant DNA technology, and also the total process of recombinant DNA technology, and then we discussed some examples of products which are produced using recombinant DNA technology. Insulin is the best example which is used to recombinant DNA technology, used by recombinant DNA technology, by which we are getting pure human insulin using recombinant DNA technology processes. In this way, vaccines are also produced using recombinant DNA technology. So before going to this video, if you were not saw the process of recombinant DNA technology and production of insulin videos, I have given these links in the description. You can go through this video and then watch this vaccine video with full of confidence. Otherwise, go through my channel's playlist and there you will find all these videos in recombinant DNA technology, means RDT playlist. Okay, let's discuss about the production of vaccines. In last video, we will discuss about vaccines are the part of pathogenic microbes. Vaccines are the part of pathogenic microbes. It may be a RNA, it may be a DNA or other protein-like substances. So, for the production of vaccine, first step is isolate the pathogenic microbes and collect those modified part or substances like RNA or DNA molecules. Collect the RNA and DNA molecules from the pathogenic microbes. So, first thing is isolate those pathogenic microbes. So, what are the steps of recombinant DNA technology? The steps of recombinant DNA technology is, first step is isolation of DNA, then fragmentation of DNA by restriction enzyme, then isolation of desired DNA fragments, then the next step is amplification using PCR, polymerase chain reaction. Then the next step is ligation of DNA fragments into a vector which is formed recombinant DNA. Then insertion of that recombinant DNA into the host cell or host organism. After transformation of this recombinant DNA into the host cell, then culturing those transgenic cells, then obtain those product and finally go for the downstream process. Then purification, extraction, etc. are used in this recombinant DNA technology process. So, first thing, in the production of vaccine, the process of recombinant DNA technology is as same as in production of vaccine. So, let's discuss one by one step for the production of vaccines. The first step is isolate the DNA or RNA of those pathogenic microbes and also isolate the plasmid vector which is work as a carrier. Then what is the second step? The second step is fragmentation of DNA by restriction enzyme. What is the function of restriction enzyme? The function of restriction enzyme is it acts as a molecular seizure, right or not? Yes. So, it acts as a molecular seizure which is help to cut the DNA or RNA molecules and also cut the that same restriction enzyme also cut the plasmid vector. And now the plasmid vector and DNA RNA molecules are fragmented. After this fragmentation, using gel electrophoresis process, separate those fragments and isolate the desired DNA fragment which is used for the production of vaccine. Now the desired DNA is separated by using gel electrophoresis process. After isolation of that desired DNA fragment, the third step is amplification. We need to amplify those fragmented molecules to make several exact copies for the more production. So in amplification, polymerase chain reaction PCR is used for amplification of that desired fragments. Now after amplification, the fourth step is ligation. What is ligation? When the plasmid vector and the desired DNA fragments caught by some restriction enzyme, that time using DNA ligase enzyme, those antigen producing gene, means those desired fragments are ligate with the plasmid vector and formed a recombinant gene. Both antigen producing gene and vector is togetherly bind and formed recombinant vector, which is carry the antigen producing gene to produce vaccine. After the formation of recombinant gene, this recombinant vector is transferred into the host cell using various transformation methods. Transformation methods like electroporation, microinjection, calcium chloride method, etc. are used for the transformation of recombinant gene into the host cell. Then culture those host cells, and for industrial production, bioreactors are used for rapid production. Now the host cells are carrying recombinant gene. Collect those cells using screening process, then extract and purified those cells. After purification, package those vaccine and send to the market, which are giving protection to all the people. So finally, we know that vaccine and insulin production process is as same as the process of recombinant DNA technology. If you will understand and clear about the process of recombinant DNA technology, you can easily understand and write about insulin and vaccine production. Let's once again revise this whole process of production of vaccine. The first step is isolate the DNA or RNA of those pathogenic microbes, and also isolate the plasmid vector, which is act as a carrier. After isolation of these plasmid and pathogenic microbes of DNA and RNA, then make the using restriction enzyme, make them fragmented. Make those plasmid vector and antigen producing gene fragmented. After fragmentation, using gel electrophoresis process, separate those fragmented and collect the desired antigen producing gene and make them amplification. Then after amplification, using polymerase chain reaction, amplification occur. Prepare several copies of those antigen producing gene for the production of vaccine. After amplification, ligation occur. What is ligation? In ligation process, using DNA ligase enzyme, in the plasmid vector, the antigen producing gene is attached using DNA ligase enzyme and make them recombinant vector or recombinant gene. Then those recombinant gene is transferred into the host cell for the production, and those host cell are produced in the bioreactor. In industries, the large bioreactors are used for culturing those microbes, those host cells, and make several copies, produced large amount of those host cells which are carry recombinant gene. After production, purified those products, extract those cells and purified them and make them, package them, and the vaccines are collect and package them and send into the market for the future use, which are beneficial for the human beings. I hope you all understand this video very well. If this video is informative for you, like and subscribe my channel. Thank you all for watching my video. Have a good day. Happy learning and keep learning.